These notes are a continuation of our biotechnology notes. So this is part 2 of our series. And in this section, I wanted to just finish up with a few other topics that are in the news, in the biotechnology realm, and kind of just let you know what's happening. To start off, CRISPR was in the news a lot and is actually kind of like the new exciting technology with gene editing. And it was in the news a lot before COVID started to dominate, bio in the news. Now, what CRISPR is, it is a gene editing tool. And what's so exciting about it is it has the ability to open up the genome anywhere. And it does that with an enzyme called the Cas9 enzyme, Cas9. This is different than typical restriction enzymes that need restriction sites. So for example, in a lab, the paper electrophoresis lab we did, our restriction site was CCGG. And the enzyme recognized that site and cut in between the CC and the GG. Cas9 does not need a recognition sequence in order to make a cut in the genome. All it needs is what's called a guide RNA molecule. And a guide RNA molecule can be made in the lab to target a very precise location in the genome and allow the enzyme to open up the genome and potentially remove sections of the DNA that a scientist wants to take out. And if they do that, for example, if there is a disease-causing gene, a scientist could remove that gene and insert a gene that does not cause disease. Or another potential application is to add genes that they desire in the offspring. So again, the benefit of CRISPR is you don't need to have a typical recognition sequence for the enzyme to work. It can open up the genome anywhere. Now, what dominated the news was a Chinese scientist who had actually used this technology to gene edit twins who essentially the trait that he edited was eliminating a gene that allows HIV to infect the body. And so these girls were born without the protein or a changed protein that allows HIV to infect cells. Now, that said, that sounds awesome. They, in theory, these girls cannot get HIV. But he essentially was not trying to cure a disease that was already occurring. He was essentially trying to prevent it. And he also, I think from what I had read in the news, he had forged some of his documents. And in the scientific community, that's kind of a no-no. And so he was sentenced to 3 years in prison. However, that just kind of gives you an idea of what the technology can do. And I'm not sure if he's still in prison or not. I have not read up on his case, but that is how CRISPR works. So to kind of continue with other applications of, genetic engineering, in theory, you could take a gene, like a pesticide gene, and put it into crops to kill insects, but make it harmless such that it doesn't hurt humans. And that does exist. There's a gene called the BT gene, and that pesticide gene will allow insects to essentially die if they eat the crop and prevent the crop from being damaged from pests. You can genetically engineer cattle to produce more milk, or leaner meat, or make cleaner wastes. You can genetically modify grains so that they are more nutrient rich. And a good example is below. It's called golden rice. And golden rice has more vitamin content compared to the wild type version. And you can genetically modify organisms to make a product for you that possibly would be needed for medicine. So this is how we actually make insulin. It's a kind of a complicated diagram on the right, but essentially what we do is we take bacteria and we insert the insulin gene into the genome for the bacteria, and the bacteria see that gene and they begin to just make insulin. So then the insulin can be processed and then packaged and given to people who have diabetes. Now, how would you do that? You actually have to open up the genome of the bacteria, insert the gene from a human that codes for insulin, and then put that gene back into bacteria, which we do. That's how insulin is made. Now, with application comes concern. There are some, concerns with genetically modifying organisms and some of the ramifications of that. And I touched upon a few here, but there are more. And so scientists kind of have to weigh the cost-benefit analysis of some of these modifications. With regards to crops and livestock, smaller farms, smaller family farms that don't necessarily have the financial means for implementing some of these genetically modified organisms, they might get put out of business. And so what we're finding, what's happening is large scale corporate agriculture is kind of taking over, and many family farms are going out of business. Another issue is just because we can change an organism's genes, should we? Again, it also can get back to ethics. This is a chicken, and this is what a typical chicken looked like in 1957, and this is what a typical chicken looks like in 2005. So because of genetic modifications, we can have chickens grow much faster and much larger. And some of the issues are they can't stand in their crates because they're growing so fast. They have health issues. And so is that okay? There's a bit of an animal welfare aspect there. There is concern that GMOs, so genetically modified organisms, their genome can escape into an untouched ecosystem. Do we want that? Do we want organisms that we have genetically manipulated and possibly, you can't control pollen. You don't know where pollen goes. Insects move pollen, wind moves pollen, rain moves pollen. And so you can't necessarily keep that contained in an ecosystem. And how do we feel about those genes possibly going to other places in the world? There is, this is anecdotal, but some people argue that food allergies have increased with genetically modified organisms. That is definitely not proven. Some scientists do claim that there's a link. Other scientists claim that there's not a link. But it is a question mark. And then finally, loss of genetic diversity. Crops are a great example. A few hundred years ago when we grew crops, we didn't have the level of modification that we do. The only thing that farmers could do was selectively breed to possibly get traits that they wanted in offspring. But now we have a lot of monocultures. And so with monocultures, and what I mean by monocultures is there's just really one genetic type of an organism, that loss of diversity causes susceptibility to disease, insects, and other selection pressures in the environment. A great example is the banana. And A couple of times in history, the banana has faced extinction because of selection pressures in the environment. Currently, the banana is undergoing a similar crisis, and it's because of disease. And because there's not a lot of diversity, genetic diversity in the environment, a disease comes through and it possibly can wipe out all of the banana crops that are out there. So again, something to think about with genetically modified organisms. And that is it for part 2 of biotech.